Biology with Dr. E.
Our next topic is vesicular transport. Vesicular transport moves large substances across membranes in structures called vesicles. Because it is an active transport mechanism, it requires energy in the form of ATP. We will focus our attention on two main types of vesicular transport: endocytosis and exocytosis. Endocytosis is transport into the cell, and there are three types that we'll talk about: phagocytosis, pinocytosis, and receptor-mediated endocytosis. Exocytosis is transport out of the cell. Endocytosis is transport into the cell and involves formation of protein-coated vesicles. It usually involves receptors and therefore can be a very selective process. So substances being pulled into the cell must be able to bind to their unique receptors. Unfortunately, some pathogens are able to hijack the receptors for transport into the cell. Once a vesicle is pulled into the cell, it may fuse with a lysosome or undergo transcytosis. This figure illustrates 6 steps in endocytosis. In step 1, a protein-coated pit in the cell's plasma membrane ingests a substance. In step 2, a protein-coated vesicle detaches from the pit and into the cell. It loses its protein coat as it moves further into the cell. In step 3, the coat proteins are recycled back to the cell's plasma membrane. In step 4, the vesicle is now an uncoated endocytic vesicle. It fuses with a sorting vesicle called an endosome. In step 5, a transport vesicle may carry some of the membrane components of the fused vesicle back to the cell membrane for recycling. In step 6, the fused vesicle may either A. fuse with a lysosome for digestion of its content, or B. deliver its content to the plasma membrane on the opposite side of the cell. If the vesicle's contents are delivered to the opposite side of the cell, this is known as transcytosis. Next, we review the different types of endocytosis. We start with phagocytosis. Phagocytosis is also referred to as cell eating. In this process, the cell engulfs a large particle by forming projecting pseudopods around the particle. The term pseudopod means false foot. The pseudopods enclose the particle within a membrane sac called a phagosome. The phagosome combines with a lysosome and its contents are digested. The vesicle has receptors capable of binding to microorganisms and solid particles. Next is pinocytosis. Pinocytosis is referred to as cell drinking. The cell takes in drops of extracellular fluid containing solutes. The extracellular fluid enters tiny vesicles. No receptors are used or required in this process, so pinocytosis is non-specific. Pinocytosis is used by some cells to sample their environment. It is also the main way in which nutrients are absorbed in the small intestine. Next is receptor-mediated endocytosis. In this process, extracellular substances bind to specific receptor proteins in the cell's plasma membrane. This process enables the cell to ingest and concentrate specific substances called ligands in protein-coated vesicles. Ligands may simply be released inside the cell, or they may combine with a lysosome to digest their contents. Receptors are recycled back to the plasma membrane in vesicles. Substances taken up by receptor-mediated endocytosis include enzymes, hormones, and some pathogens. The second type of vesicular transport is exocytosis. Exocytosis is a process of ejecting material from the cell. The substances being ejected are usually packaged in secretory vesicles. The vesicles contain proteins called V-snares, while the cell membrane has proteins called T snares. The two proteins interact and trigger exocytosis. Substances removed from the cell by exocytosis include hormones, neurotransmitters, mucus, and cellular waste. This figure illustrates the 4 key steps in exocytosis. In step 1, a membrane-bound vesicle is shown in the cell's cytoplasm. The vesicle contains molecules to be secreted from the cell. The vesicle surface has membrane proteins called V snares. This vesicle is migrating with its content to the plasma membrane. In step 2, the secretive vesicle is at the plasma membrane. The proteins on the surface called V snares bind with membrane proteins called T snares. In step 3, the vesicle and the plasma membrane fuse, forming a fusion pore that opens into the extracellular fluid. In step 4, the vesicle contents are released to the cell exterior through the fusion pore.